Welcome all to the next version of the webinar series of X-omics, and it's a collaboration with BBMRI. This is the third in a row, and we have one more to go. It's the last one is actually your chance to pitch your own project, so please take your chance and do that. I think for today we have a very exciting lineup. And just before we start with the first talk, I would like to ask you to ask your questions after all of the talks. So we have a talk of like 25 minutes or half an hour, and then we have a chance to ask questions. You can do that by raising your hand or typing it into the chat so that I can read it. So without further ado, I would like to announce the first speaker, who is that is Patrick Deelen from the University Medical School of Groningen. He will talk about using RNA-seq and genetics to identify core genes. Please go ahead, Patrick.
Oh, thank you. Very happy to be here. So today I would like to talk to you about different projects that we are doing to actually find both rare and common disease genes. And I would first like to start with the rare diseases. And yes, I started the correct slides. So, and I would like to start with the point that our knowledge of genes is tremendously biased. So there are a few genes, for instance TP53, which is a very famous oncogene, for which a lot is known. And there are actually thousands of papers dedicated to describing or studying the function of this TP53 gene. But on the other side of the spectrum, there are also thousands of genes for which we know nothing. And the majority of these are non-coding genes, but there are also a lot of protein coding genes, which are simply never studied in detail. And that's of course a problem if you want to study the genetics of diseases, because these are just as likely to happen within these well-studied genes as in other genes. And to do that, we make a lot of use of a data set accompanying over 30,000 RNA sequencing samples that we downloaded from the public domain. And this is a very diverse data set. So we did a UMAP on this, and as you can see, we have here a cluster of all different types of blood cell types. On top, we have different kinds of cell lines, and we have some primary tissues over here. You can actually zoom in a bit so that you can also discriminate between these different primary tissues. And we have a bit of an hematopoietic signal all the way on to the right. So there's a lot of biology described within this data set. And we use this as follows, and this is just a very cartoon-like way to show this, but suppose you have 3 genes, and these 3 genes are known dilated cardiomyopathy genes, or DCM genes, so they cause this malfunction in your heart muscles. And then we can take our 30,000 RNA sequencing samples, and we can basically calculate co-regulation. So to what extent are these genes regulated in the same manner when looking at its expression levels? And then we see that these genes are very strongly co-regulated. This is actually an older observation that genes with a similar function are very often showing the same expression patterns. What we then can do is we can take another gene, for instance this TP53 gene again, and see if it is co-regulated to these known DCM genes, which it's not. So this would not be a very strong candidate to cause a DCM. On the other hand, if we take, for instance, this OBCSN gene, and we look at the co-regulation there, we see that this is strongly co-regulated to the DCM genes, making it a very viable candidate to also cause a DCM phenotype should it be damaged by a pathogenic variant. We do this by looking at human phenotype ontology, and that is a way to describe human rare disease phenotypes, or actually human disease phenotypes, you can generalize it. And for each of these terms, there are a number of genes annotated to it. And it can be a very general term, like abnormality of the skeletal system, where there are more than 1,000 genes annotated, or a very specific type of hip dislocation, where there are only a few genes annotated to it. And for each of these terms, we can predict the genes that are associated to this based on the currently known genes. And if we would do this for the dilated cardiomyopathies, if we do this in general, we're going to get for all 50,000 genes, protein coding and non-coding, and for all pathways and HPO terms that we put into our predictions, we're going to get a prediction score. And here in the histogram, we show the dilated cardiomyopathies again. And in gray, we have the background distributions of genes that are not yet associated to DCM. And in green, we have the distribution of the known DCM genes. And it's not very visible because it's a density plot, but there's actually a bit of a tail here. So there are a few dozen genes here in the tail that also get a very high score. And one of these is this OBCSN gene that I already mentioned before, and I will get back to this gene later. And the idea that we then have is how can we use this in a diagnostic setting, or at least in a rare disease research setting, that if you have patient-specific input, so for instance the HPO terms that you observe for this patient, and you have a set of usually about 250 genes that have a potentially disease-causing variant. So going through each of these 250 genes for each individual patient is of course a lot of work. So there we devised the GADO method that uses our gene network predictions based on these 30,000 RNA sequencing samples. And then we can prioritize the genes based on the HPO terms that we specified for this patient. And the essential idea is that you're going to start with the biggest marble first. This is something for which we have a command line tool if you would like to run it on your own cluster, but you can also do this on our genetwork.nl website. We can click on HPO gene prioritization. You can click on a few genes, plug in a few HPO terms, and an optional is that you also put in your candidate genes for which there are variants. So here it's nice to know that you don't actually need to specify the specific variant, just which genes have a variant, making it very privacy friendly. And after you have done that, you can click prioritize, and then you're going to get this ranking. Well, in this case, it was a patient with Kleefstra syndrome, and for which actually the diagnosis was already known. But here our GADO algorithm prioritizes this gene on the second place. Despite the fact that there are actually two HPO terms here that are not associated to Kleefstra syndrome, so actually another reason why this person has that, and that this is only a few of the symptoms that are normally described for Kleefstra syndrome, we can still find back these genes. But you can also, of course, apply this to unsolved cases, which is tremendously more interesting. And then we come back to this OBCSN gene. This was a patient with a dilated cardiomyopathy, and within the subset of genes that contain a putative causal variant, on the third place we prioritized this OBCSN gene. And then later we had a closer look at our cohort that we had, and we found a second patient also with a similar mutation in the OBCSN gene. And in literature, we found one paper also describing a dilated cardiomyopathy or another cardiomyopathy phenotype for this gene. So it was not yet in the databases like OMIM that is typically used for rare diseases. It was also not in our training set. So basically we came here independently of this already existing paper. And that brings me also back to this distribution where you can see that this gene is really in the tail of our prediction scores. Then you can also use the gene network website to have a closer look on what's going on. For instance, we can simply type in this gene and we can see that it's already annotated to sarcomere organization, which is a process very important to a proper cardiac function. But we also predict some other biological processes for which it's not yet been annotated. We can then have a look at the known DCM gene and then also plug in the OBCSN gene in this plot. And if we zoom in a bit, we see that this OBCSN gene is closely related to these other blue genes, which are made by an automated clustering. And this subset of OBCSN genes are very important for cardiac muscle tissue morphogenesis. So this already gives you some clue, not only that this gene is being prioritized, but also why it's being prioritized for this specific phenotype, how it could be relevant for your patient. So when we did this in practice, we used a cohort of 83 solved cases. And on average, when we made a selection for protein coding for actually known disease causing, here we had a selection on average 56 variants in known disease causing genes. And so that would be a more clinical relevant setting. And in 42% of these cases, we would actually put the Colso variant in the top 3. So then you would, for a majority of the cases, you would only have to look at the top 3 variants already coming to your diagnosis. Then we also applied it to a cohort of 61 cases which are not solved by routine diagnostics, and we found strong candidate genes for 10 of these cases. And due to a stringent cutoff, on average we only had to look at 2.9 genes per case. And the burden of evidence that we have varies, but we actually have one gene which was completely novel at the time, that we also validated using experiments. And there's now also some other papers with patients where they found this variant. And the OBCSN gene is among these where we have now multiple patients, which is a very strong candidate. So we think this is a nice way to look at unsolved patients and prioritize candidate genes. But up to now, I've been talking about rare diseases. And one of the topics of this talk, and something that we're active with now, is how can we link this to the common diseases? And but these rare Mendelian disorders, it's typically one variant within a single gene, at least a single gene within a patient, and then you get a specific phenotype. These common traits and diseases are usually caused by a very large compendium of many different small effect variants, and they might typically they can quite often they can be related to a close nearby gene. But how this then influences the phenotype is typically unknown. So we don't really know how these different genes work together to cause a specific phenotype or phenotypic variation. So what we are doing is we are using GWAS summary statistics. And using these GWAS summary statistics, we're going to go from variant p-values to gene p-values using an existing Pascal method. After that, we're going to use our co-regulation data, again from these 30,000 RNA sequencing samples, and we're going to try to capture which genes are now very often co-expressed with the genes nearby these GWAS hits, so the genes with a very small gene p-value. And when we do this, we actually come to some prioritized genes, and they might be core genes, as a discussion more in the common disease field. Up to now, we typically use the term key genes to be a bit more safer from that discussion, but genes that we at least predict to be more relevant and more informative for the studied phenotype. And when we do this and we look at different traits, and we see from, well, which key genes do we now prioritize? We have a set of almost 160 key genes for these 25 different traits. And if we then look at the loss of function intolerance score, so basically, if you get a loss of function mutation in this gene, is this going to be compatible with life? And this is a score calculated by the CHRONOMAT Consortium, where they have, for tens of thousands of people, have exome sequencing and whole genome sequencing data. And what we then see is that a very large chunk of our predicted key genes are completely intolerant for loss of function alleles. Even more so than what you would see if you would just look at Mendelian disease genes. And again, much more than, of course, a background gene of background set of all the other genes. Indicating that these genes are very important in your biological functions and are conserved by evolution. And to give you a practical example on how this then look like, when we look at inflammatory bowel disease, and we have all our cis genes, so basically genes within the GWAS loci, those are colored green here, and we have the key genes, so the genes that we predict to be important. And as you can see, we then get all kinds of connections from all these cis genes that are converging on these key genes. And if we then highlight one chunk, one of the top core genes that we predict for inflammatory bowel disease is NF-κB1, which is actually a known driver gene of IBD, but it's not found within the GWAS. So it's very nice that we can indirectly find this gene back. And it has a direct, strong co-expression with a few genes within the cis loci, which are in green here, like TNF and CSF2, but also with a whole bunch of other genes that are predicted to be predicted to be important in IBD development. And what we then can do is we can actually take all these IBD key gene scores, and we're going to test how well are they able to predict known rare disease genes. For instance, the top AUC, so the prediction performance area under the curve. And if you would have a 1, you would have a perfect prediction. If you would have 0.5, it would basically be tossing a coin. And here we see that these IBD genes are actually very well able to predict rare disease genes where people are susceptible to candida infections or hypersensitivity of the skin and all kinds of other infectious and immune rare disease phenotypes. So to summarize that, we can go, we can go from these GWAS cis genes within the GWAS loci. We can predict genes outside the GWAS loci, which are currently not being found with GWAS. So they're acting in trans. And these genes are very strongly depleted for loss of function alleles. They're strongly enriched for rare disease genes. And thereby they can also be used to predict new novel rare disease genes. So up to now, we have been using these 30,000 samples that we downloaded already a few years ago. But we're now also making tissue-specific networks. For instance, something we call kidney network. The preprint of this will go online in a few days, where we downloaded and accumulated from different sources kidney RNA sequencing data. Again, you can make a UMAP and you can nicely discriminate the different kidney regions. And then we can also calculate these co-regulation scores. And what we then see is that when we look at a set of 99 kidney-related HPO terms, that for a quarter of those we are having an improved performance by using this kidney network. And here you have to realize that this kidney network is only about 700 samples compared to these 30,000. And what we then see is we look, if we use that data, we see that the genes that are currently known for, in this case, hypomagnesemia, we see that many of these edges are actually stronger present within the kidney network, and many edges are also not present at all within the gene network of 30,000 samples. And this also gives an improvement of the AUC value from 0.84 to 0.92. Well, that might not sound like a lot, but that actually gives you much better prediction performance in practice. So one example that we can then do is we can use a GADO method again. We can plug in HPO terms. We can take the exome sequencing data of the patients, and then we have candidate genes based on whether or not they are predicted to be relevant for this phenotype, and we have candidate genes based on presence of rare variants. And then we were left with 3 genes which are overlapping between these two, and then one of them is ALG6. So I'm not the person to go in detail about the biology of this ALG6, but we have a collaboration with UMCU Utrecht. And the last thing what I want to talk about is for future improvements that we're working on, and that's actually based on some things that we already did a few years ago. And there we found that if we look at an EQTL for FADS2, so we have here this FADS2 on the y-axis, and you can see by these 3 different lines that there's an EQTL effect. On average, people with the AA genotype have much higher FADS2 expression than people with the GG phenotype. But we also see that this EQTL is its strongest when people have a low SRAP-F2 expression, and that the EQTL effects get weaker if your SRAP-F2 expression is actually increasing. So then it matters less and less what kind of genotype you have. In this case, that makes perfect sense, because if we look at the FADS2 locus that we see over here, this variant for the EQTL is mapping within a binding peak of SRAP-F2. And these two things combined make actually such a plot very plausible, where we have this equigel effect of RS9685667 on FATS2. So this is a genetic regulator, but this X, because SREP-F2 is actually binding there with a different affinity, and that is actually a likely regulator of FATS2. So if we would then trying to make a network, we can actually make a directed network by integrating this expression in genetic data. So in this case, we have this genetic variant, we have the expression of SREP-F2 and FATS2. And instead of just plainly looking at co-expression linking these two genes together, we can actually draw an arrow there between the two different genes. So one of the projects that we're currently starting up is doing this at a larger scale. So hopefully that we don't have to look simply at co-regulation in general, but that we can actually use these directed networks to make these predictions even better. And this is already a plot now of almost 2 years old. We see that the available data within the publicly available RNA sequencing data in the European Nucleotide Archive is actually growing exponentially. So these 30,000 samples that we use now, that's actually something downloaded midway 2016, and half of these passed the quality control, so the line would be somewhere over here. But with the current growth, we're actually over 1 million samples at the moment, so we hope to be able to doing this in an increasingly larger scale. And with that, I would like to end this broad overview of the different things we do integrating rare and common disease genetics with expression data by acknowledging all the different people that have worked on this or are working on this.
Yeah, great. Thank you, Patrick, for this very nice talk and overview. So are there any questions for Patrick?
I see Victor raising his hand.
Okay, Victor, please go ahead.
Well, Patrick, thank you for this excellent talk. It's really inspiring. I have a question with regards to the input of GWAS variants. So what we try to do is to figure out what the modifiers are for known disease genes, but the actual cohort that we have is way too small to do anything like GWAS on. So could you recommend on how to go forward with the GADO system then?
I'm not sure if I completely follow what you're doing. So you have rare disease patients and you want to use common variants to look for modifiers of this rare disease. That's what you're doing. Okay, that's a slightly different thing than we have done so far with the GADO system. But I can imagine that if you, for instance, have a phenotype that is relevant for this rare disease, you could for each gene predict whether or not it's relevant for this phenotype. So you could use this as a prior for your GWAS, that you're going to specifically study genes that are predicted to be relevant for your phenotype of interest, so that you can actually limit the multiple testing burden. So that you say, I'm only going to do a GWAS there. That's the first thing that comes to my mind when you ask that question.
This is a very good answer. Yes. Thank you.
You're welcome.
Any other question for Patrick? I have a question, Patrick. So the GADO tool and gene network, is it the same? No, it's two tools, right? So
no, so the gene network is basically just the data and the function predictions, and the GADO is a tool that uses the network data to actually prioritize genes for rare disease patients.
Okay, so and is it freely available? So can you just upload your own data there? Or
you can upload all your own data on genetwork.nl, and there we have the GADO running and all the gene function predictions. But we also have a command line version, and you can download all the data. Everything is available, and if you're missing a file, you can always ask, and we will make it available. So this is completely open.
Okay. And I have another question about the kidney network that you showed. Yeah. Was it based on single cell data, or because it looked a bit like this?
No, we use the UMAP, so then indeed people often associate it to single cell data, but it works just as well for bulk RNA sequencing data.
Okay.
This is one of our future dreams, to also build single cell networks, but that's a bit further up on the roadmap.
Okay. And so you could build your own sort of network if you use your methodology for your own target tissue, then basically if you have the data.
Yeah, also the tool to create the networks is fully available.
Yeah,
and documented.
Okay. That's really nice. Yeah. One more last question I have. There's a question here in the chat. Can one use gene network for analysis of genes derived from proteomics data?
Well, that's actually a very interesting question. So in principle, as soon as you have data that you can calculate correlations between genes, you can use gene network. And we have a project going where we use methylation data to predict gene functions. So we first linked the methylation from the BBMRI project, it's the BEOS project, to the genes using gene expression information as well. And then we use actually the co-methylation score between genes to predict gene functions, and that works surprisingly well too. So I think proteomics data can be used should you have this on a sufficiently large scale.
Yeah, okay. So one question is the directed networks. Is that trans EQTLs or not? I didn't completely understand how that was built.
No, it's built on cis EQTLs.
Okay.
Can I go back? Yeah. So actually we have this genetic variant here on the left bottom. That's just a cis EQTL of FADS2. And SREP-F2 is a trans-acting factor. So that is coming from somewhere else, but it's just modulating the cis EQTL effect. So in principle, of course, this can be extended to trans EQTLs, because as soon as you can fill in these other things here on the bottom right, it's very reasonable to assume that this genetic variant via this directed network is going to be a trans EQTL for this 13 over here.
And you have this SREBF2 because it's co-expressed with FADS2. Is that why
It's also co-expressed, but actually this was a genome-wide scan for modulators.
Okay. 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 Well, if there are no more questions.
One more question. In the chat popping up. Would the result of your tool be more reliable when using pathway enrichments? Yes, actually, the pathway enrichments are more easy to predict than human phenotypes, because what we see for human phenotypes very often is that there are multiple pathways and thereby also unrelated genes that can cause the same phenotype should one of these two pathways be damaged. But if you would want to use it in a clinical setting, then you would first have to really know which pathways are going to be relevant for which phenotypes. So that's a challenge we did not solve yet.
Okay. Well, thank you again, Patrick.
Very welcome.
I think we move on to the next speaker, which is Gennady Roshchupkin from ErasmusMC. Gennady is going to speak about the use of artificial intelligence in multi-omic data. Please go ahead, Jenna.
Yes, thank you. Can you see my screen? Can you hear me? Yes. First of all, thank you for the opportunity to present today. And I'm really happy that so many people excited about this topic. So now on this workshop, I'm really excited myself. So I hope that it will be interesting, and I hope for questions afterwards, because just please remember there is no stupid questions, only stupid answers. So you just please ask if something not clear or you want to know more. Of course, there are a lot of literature about this topic nowadays, and I try to make in this presentation some summary Of course, I might be biased because I'm working in this field myself, but I know that the slides will be shared afterwards, so you can check the references and some links I also provide at the end. So, and yes, as I stated in the title, so I think that's a good definition, like how this artificial intelligence can improve, let's say, make our analysis faster, better, stronger. But first, because well, I'm not sure about knowledge of the audience, I just want to make first some intro to the artificial intelligence and the field. So of course, artificial intelligence by itself sounds really exciting and promising and some way futuristic, but we should understand that, well, when you hear someone talks about artificial intelligence, it most probably it's about machine learning algorithms or about deep learning. So in deep learning, it's about neural networks, which consist of the neurons. And if you simplify it to the one unit, so basically one neuron is just, it can be just basically linear regression. So you have some input parameters, you have some output function, and in simple case, one neuron can be just represent a linear regression, or if you change it a little bit, it can represent just logistic regression. So, and that from one hand, this is quite simplistic, but from other hand, this allows you to build really complex models. So let's say if you want to connect many neurons and many layers together and input some data, you can see how they activate and how the data propagated through your neural network. And that gives you, based on the, you can make it bigger, you can make it wider, etc. So I will mention later a few examples. And this is example, it's like one-dimensional data input, but of course you can use two-dimensional data. This is just a nice example where you have a handwritten digits, so you have a 2D image, and then every pixel of this image is the input parameter to neural network, and you can see how that propagates through these layers, and some neurons are activated. So basically it's a combination of many linear regressions or logistic regressions, and at the end it can allow you to analyze really complex data and perform analysis really well. And of course there are different types of architecture, as I said, so I will cover a few examples which I think are really promising and interesting in the field, and I hope that in the coming years will be more and more research based on these networks. But you can see that there's like actually quite a zoo of different architectures. Some of them just really simple with one layer, some have more layers, some have some convolutions, some really have end-to-end connection, or let's say the last example, RNN, it has some memory even unit. I'm not going deep in details. That one is mostly used, let's say, in some language processing analysis. But coming to the topic of this talk, so why it's faster? So, and why deep learning? So, and it's quite simple explanation, because we all know that, especially with omics data, we are having every year and more data available, and we need better and better algorithms to analyze this. And you see that actually deep learning allows to analyze such data much faster compared to the classical algorithms. And why this is the case? It's kind of I would say positive loop, because it started that deep learning can be simplified to these simple units like neural networks, neurons, and the simple operations of the matrix, basically multiplication or vector multiplication. And the GPU cards were really good for this. And once it became obvious that GPU card can speed up this process, then companies, in this example, NVIDIA, they started to develop better and better GPU cards for such type of the computation. And that actually even accelerated even more the performance of the deep learning, and then it forced companies to develop even better GPU cards and etc. So and then that's why deep learning can be really fast. And you can see it as examples for the image processing, so that there are so many algorithms in different type of medical image analysis, and there are a lot of companies which already registered their AI or deep learning based algorithms, because again, so that gives really high performance for medical image analysis. But if you think about the application for the fast omics analysis, so the first, I think that the most obvious example and which came in mind, that's about imputation of the data. And I made some summary of the algorithms where they can be used. It's about genotype data imputation, it's about gene expression imputation, about epigenomic data, proteomics. I haven't mentioned it here. Again, so just remind that the slides will be shared afterwards. You can check these tools if you're interested in. So, and just I want to focus your attention that a lot of methods are based on the so-called autoencoders. So I will cover this later. And if you really want to go deeper in details with this review, I highly recommend this really nice paper. You see reference on the bottom. Yeah, so faster. So it means that GPU cards and the deep learning computation brings it to the next level where we can just save the computational time on the scale from basically from years to hours and days, because on GPU computation can be really fast. And well, in principle, GPU cards can be really cheap. Well, some of them are really fancy and might be expensive, but nowadays to perform some simple experiments, it's not necessary to buy a really fancy GPU card. So what about better? So why that type of the algorithm becomes so popular and outperform state-of-the-art methods? Well, but that's, in my opinion, that's simply because there is a mathematical evidence, which is just called universal approximation theorem, which says that neural network with actually even a single layer is able to approximate any function in the universe, basically. Only one point, of course, that we don't know how many layers and the architecture of this neural network. That's why, as I said before, you have so many different types of the neural networks, each of them trying to find this best solution for your specific case. And therefore, if you think about this machine learning or deep learning applications, so basically, if you have any of these low-level problems like clustering, dimensionality reduction, classification, or regression analysis, here you can solve this problem based on this theorem in the best way with the deep learning or neural networks. And as I mentioned that in the imputation part, that this autoencoder, that's one of the type of the neural network which I want to focus more your attention in this presentation. So this idea is quite simple. This is example again, so with the hand written digits, where you have some really noisy data. And this network, what it does, it just first reduces the dimensionality towards like latent space. So we'll say from like hundreds of input parameters to just a few or tens of them in latent space, and then tries to reconstruct it in the decoder part to the original image. And by some manipulating of the training, during the training and some parameters, you can actually achieve that autoencoder learns just a global pattern, and that's why your output will be denoised in this way. And the same type of the architecture using you widely use now based on the examples I showed you for the imputation of the data, because once you train it, it can impute or denoise your data really fast, like tens of times of magnitude faster than classical statistical method. And this is just this is example with actually denoising for genotype data. As you see that, for instance, in input you have missing values, and as output you have already imputed data. So the accuracy of such method is quite high, and the speed of this such type of algorithms also, as I mentioned, due to the GPU computation and such type of the optimizations is really fast as well. Another example is this is imputation for specific location. So this is a really nice paper. I think this was published recently. This is bioRxiv link. That's about the multi-etic actually imputation, because you know that the genotype information is might vary, and if you train the algorithm to impute the data, then you can overlap the specific region in this case to perform the trans-etic analysis. It's really also interesting application, and I suggest also the paper to read if you have a similar problem. Another example is actually with this gene expression. So again, so as I said, it can be used for denoising, but also here, there's a nice example that you can actually manipulate this latent space and force the algorithm to learn specific pattern. So, and as well, because I know that I think that people working with this type of the data know that the data can be quite noisy. This type of the pipeline can be used to denoise this data. And it's again based on that idea of the autoencoder neural network. Another example, if you use a methylation data, so you can not only train for denoising, but you actually can use this latent space, which is much smaller than your input parameter, to explore your data and also apply the method like t-SNE or UMAP to see that does your network capture a specific pattern. So in this example, with methylation data, the latent space represents, let's say, healthy clusters of healthy people with cancer. And what is interesting that using the method for explanation of your prediction accuracy, I will mention this method sharp later in the presentation. So you can actually see which of the CPGs are important for this clustering. So you can go back to your raw data and see that some CPGs are more contribute to the healthy subject and some of them to the cancer. So that's also interesting application of the autoencoders. Another actual type of the neural networks, which I also found really interesting, it's a new type of the networks for survival analysis, where you can integrate the multi-omics data as well for basically Cox regression, but that type of the network, they can approximate even the interaction between parameters. And let's say if you're also working with imaging data, then you can combine together such networks without any pre-feature selections. And if I can recommend a few really good examples of the pipelines and papers, it's quite recent papers about spatial single cell analysis, so which covers really amazingly like the whole pipeline written in Python from raw data to some interactive visualization and statistics, and also availability to run deep learning methods. So this is one example, and another example is EMAP. That's a bit more busy and complex picture for the single cell dataset analysis, but basically what it does is also does denoising and also batch effect correction. So again, so just check the slides later. You can see the reference and check the paper in more details. But now coming back to the more topic which I myself interested the most, that's about the problem with the black box. That's probably most of you heard about artificial intelligence, that it's not explainable, and the problem is especially in the medical application, healthcare, in the omics analysis, that you want to know why you achieve certain performance, and there are actually, it's not anymore really black box, because there are a lot of development around. And one example, as I said, is Sharp library, which basically brings the model from black box to explainable algorithms. It applies for machine learning and deep learning methods. There's a nice also paper with application for the electronic health records data, so where you can see which actually data point and parameter are the most relevant for your prediction. But of course, we want to apply it for omics data. And there was like a few years ago, a nice review that how we can actually, based on our knowledge, already biological knowledge, how we can improve and make a new type of the neural networks. And that's GeneNet framework example, which I'm myself involved. And this paper you can also find on BioHive. And this is a really interesting idea, how you can integrate biological knowledge. So basically you start from genotype data, so that's data you need for this network, but the rest are built on the public available data. So you can integrate knowledge about how these SNPs are connected to genes. Then you have a gene layer where each node represents just gene. Then you can connect these genes to the pathways. And actually that's the whole library integrated in this pipeline. So from the GTEx, Allen Brain Institute, Kecker pathway annotation. And actually that I was thinking when I was in the first presentation today, I was thinking that actually that network information was really interesting, that it's also possible to integrate in such network architecture. And at the end, so basically after you trained, you can reverse the network and see, for instance, which layer are the most important for your prediction. So basically you can get the gene-based Manhattan plot. Or if you integrated information about that you connected your neurons, that they represent a specific cell types, you can see which cell types enriched and the most important for the prediction. And of course, that such ideas can be used not only for genotype data, for microbiome data. And as I said, our idea, of course, a bit futuristic, that to be able to build something like this, but of course, we're still not yet there. But for instance, example, which I wanted to mention, that we are working together with BBMRI and actually Joyce, that in multi-omics analysis, where we're building similar neural networks, but where we integrate gene expression, methylation data, and also you can even integrate some covariates and correct your analysis, and then see how this covariates integrates with your interact with your methylation data and gene expression data. So to summarize about beta, so that the analysis can be more accurate, based that the neural network can basically approximate any function, then the neural networks can capture more complex pattern. And nowadays there are a lot of algorithms and development that to make them interpretable. And the last part I wanted to talk about is about making it stronger, because we can say that, yes, so that all these algorithms are really nice, but we know that the effects are really small and we want to detect them. And normally we do this huge consortium-based analysis with the meta-analysis, and we can't share raw data. And here comes another project which I'm also excited and involved in my own research, which calls the federated learning. So that's basically a classical scheme for the GWAS meta-analysis, when you have a central site and then you collect data from another cohorts based on the summary statistics. But with deep learning analysis, nowadays, it's possible to do the same using specific protocol and frameworks, where the network, what I showed you before, will be integrated in local cohort, but the data will not be shared. It's only will be shared some parameters over the network, and then and then the results will represent basically like a pooled analysis without sharing the raw data. And idea is quite simple. There are different ways to do this. So you have a central site. You can see this workflow that you can distribute your model to other sites. They run the local analysis, then they share to the central site, and then central site aggregate the model results, and you just keep repeating. So basically, you just need a good internet connection, which nowadays is not really the problem. Or you can even run it in the peer-to-peer model, so where there is no central site, everyone aggregates everything, and then at the end you just summarize the network. So that's more about again, so some details of the implementation and about the, let's say, the computational burden for the internet connection. but at the end you will get the result the same way as you have all data in one place. So basically pooled analysis without sharing raw data. And there are quite some development in the deep learning community. This is example of the open source framework to do this, mostly focused on the image analysis. And there's a special Python library to do this. And the companies involved in such analysis, let's say NVIDIA, they have a pipeline for the image analysis again. But we are collaborating nowadays with the NVIDIA to develop this pipeline for omics analysis, for omics distributed analysis. And to summarize, so that basically that application new method can accelerate our research, while it can make us stronger. So also developing a new method based on the expertise and data. So that I think the role of community to bring knowledge to the artificial intelligence or deep learning frameworks, and that can not only accelerate but also give more knowledge and insight in the analysis. And of course, distributed learning will change the way how we do collaboration and work within consortiums. I just, if you want to recommend the frameworks, of course, nowadays all this deep learning mostly done in the using Python language, and this is the most famous frameworks to do this. Again, you can check slides later. If you can just use some time for self-PR, just please, if you're interested, for instance, in the microbiome data. So we just published recently within COST ML for microbiome action review paper. And of course, also we are doing a lot of in the another COST action consortium network. So Gemstone, and also within Charge Consortium. So if you're interested, you can always join and read our also results of our research. And I also leave a few links, so you can check it later, and you can play around with some deep learning and machine learning, just to give impression if you don't have any knowledge. So it can be a good start to understand the intuition behind these methods. And with this, I want to thank everyone. And please ask your questions. As I said in the beginning, there is no stupid questions, there are stupid answers. Thank you.
Thank you very much, Jenna, for your very nice talk and overview of all of the methods for AI. Please, if you have any question, just raise your hand or type it into the chat. So for me, I'm a biologist from background, and I'm always very impressed with all these computer tools. So these last links that you sort of gave, that is a good starting point, but is it always, when you want to do this kind of stuff, do you really need a lot of background for this AI analysis?
Well, I think nowadays it's really simple. So you need to know, I can't say that you don't need to know anything, but definitely you need to know to have some scripting examples and about program languages. So, but nowadays with the libraries I mentioned about, especially the one, the Keras, it's really, so it's really easy to run a simple analysis. Of course, if that doesn't work and you need to go deep in details, that that can be an issue if you don't have experience enough with this. And another thing, if, well, actually the most of this analysis nowadays is like 80% is about data cleaning and QC and preparing your data for analysis. Because if you compare that the how many lines you need to write some neural network, it may be like, sometimes it's 5 lines of the code or 10 lines of the code, but then it takes like 100 lines of the code to prepare your data for the input. So Yeah, that's
quality of the data, of course, is very important. There are several questions. So to begin with the first, is this modeling affected by the degree of dynamics, such as intransigent proteomics and metabolomics?
Just to make it clear, it means that, just to understand the question better, so it means that this data is really dynamic, and that's why if you combine together multi-omics, let's say like gene expression with more dynamic, or like genotype data with more dynamic data like proteomics, can introduce some bias. Yeah, I think that's, but that's the same as for any type of analysis. Let's say we wish to have a data, especially for dynamic, like proteomics and metabolomics, like measured, I don't know, like every hour based on some device from your finger, I don't know, but that's not possible, of course. And I think that I actually think that such type of the method can capture the pattern if there is some global pattern. So let's say to remove the bias and noise, this noise of these dynamic structures, better than the classical statistical method.
Yeah, but I can imagine that also here it is important to choose your data right. So if you have your target tissue, then only use your target tissue and not all the other data. Maybe that's also important, because dynamics, of course, in ours, but also dynamic in different tissues, for example.
Yeah, but for instance, that the network or architecture with these explainable nodes, for instance, what is interesting about this type of the analysis, so if you do some pathway enrichment analysis, classical based on GWAS, then you have to correct by number of pathways you tested. So basically you can even overkill your results based on this. But in a neural network, if you add more neurons which represent some pathways, so if they are not relevant for the analysis, they will be just left out, basically. So in this way, the power can be higher, so you can actually pull as much information as you want, and then the idea that the network can sort out which one the most important.
Okay, so question, how can we use TCGA cancer data sets like DNA methylation, gene expression and copy number variation data combined together in deep neural networks models?
Well, I think that relates to the project what I mentioned, that we are working actually on in the BBMRI and together with Joyce about this multi-omics integration. So basically, I think that maybe the good example about that in neural network, you can have easily different type of the channels as input channels. Let's say if you analyze the image 2D image, you also have RGB channel for different colors. So, and the same approach can be used in neural networks, that you can just use a different type of the data as a different channel, and then it's actually up to you to decide, do you want them to interact from the beginning, or you want to first, let's say, go deeper with your layers and only allow them interact on the, let's say, on the high level of the features extracted from these data sets. So I think, but of course you need quite some data or outcome to do this. I think that if it's for the cancer, I think that's actually a good example where you can much easier to detect the difference between case and control than compare, let's say, to just a classical continuous trait of the human. Yeah, so I would say that it's just with a different channels approach.
Okay, so next question is, could these autoencoders be used to solve common problems like missing data imputation?
Yes, that's definitely, that's actually a few examples I showed that now used a lot. So, and yeah, that's I think that one of the ways to go is, like, if you have some problem about this, so I think that autoencoder with missing data, that would be the way to go.
Okay, and the last question I see in the chat is a very long one. Yeah, but maybe I missed it, but what is the advantage to perform GWAS MA with deep learning networks as compared to combining summary statistics from a standard linear or logistic regression? So he's asking about statistical advantages. Would it be a better model fit?
Yeah, I think that, well, in the first approximation, let's say that if you have the deep learning networks, and well, that's a good question, and really maybe a long answer. I think that the advantages could be in a few things. Okay, first that well, deep learning by definition is better than, by like a theoretical definition, is better than linear regression, logistic regression. Of course, second that it can model interactions. So if you assume there is some epistasis for genotype data, and definitely for like metabolomics and proteomics and I know other omics, there is definitely interaction, and that's network can model this better. And yeah, and I think that in a way in the future that will replace the PRS approaches for polygenetic risk scores, because that can be just more accurate, but that will require, let's say, just a larger data set to train the networks.
Okay, so, and I think the last question from Ingrid, are you familiar with the H2O AI platform? And if yes, how does it compare to the platforms you are using?
I'm not sure. I definitely heard about this platform, but I think that's some high-level framework. So basically, they trying to make it easier to build a network. So I think that if I'm not mistaken, so that's the main difference, is that it gives you easy to start, but then if you need to change something, it might need you can't do this basically. So I think that if you let's say to learn from scratch like Python, let's say, that's a bit higher input, like learning curve, but then you will be more flexible, and then with such frameworks.
Okay, well, thanks again, Jenna, for your nice overview. I think we will move on to the last speaker of today, which will be Erik from Akker, who will give a talk about metabolomics-based predictors and how to build them. The floor is yours, Erik.
Okay. Well, thank you for the invitation to present here on this webinar. Thank you also for coming here. It's a very large audience. Nice. So my presentation will be a bit more epidemiological style. So this is about how to build metabolomics-based predictors as markers of health and disease. I'm at the LUMC and TU Delft. I work there in the Leiden Computational Biology Center. I work a lot also with the sections molecular epidemiology with Eline Slagboom. So what is, I think, this. So what is the aim of this research? So what we try to achieve in aging research is to find good, suitable markers for biological age. And biological age is basically the discrepancy between your actual age and the age you are perceived to be. So to explain, by this, this is a picture taken from this nice review. So here you have chronological age. So people at a particular age can actually look very different in terms of how well they are doing. So it seems that in some people, this aging is more rapidly happening than in other people. And we would like to find out if we can actually score this difference, this discrepancy. And we use omics predictors. So I think in that sense, these omics predictors are very much like a mirror. So if you would look in the mirror and you would see that you would look way older than you actually are, then this would be a bad sign in terms of health, right? Everybody who looks older than they actually are, that doesn't really sound good. So what you also can do is look into omics data, predict your age, and see if you actually are look older in your omics data than you actually are, as a marker of biological age. So to explain a bit more, yeah, yes. So I've taken, so what they call these algorithms, they call them clocks. So basically, these are considered to be molecular clocks to see how fast you are aging. And I've taken here an overview picture from another paper where basically the rationale is then behind training such predictors. So in this instance, and most famously used, are the methylation clocks. So especially Steve Horvath popularized the methylation age and the use of this. So what you do is you use, for instance, an omics resource, you use some type of model, in this case, an elastic net regression, you predict age, and you take some measure to show the discrepancy. You either divide it or subtract your actual age from your predicted age. And what you then can do, you can follow up with all kinds of clinical trace to see if people who are estimated older than they actually are also have a higher risk for future incidents of particular outcomes. So again, how this looks like, you predict age. Some people actually look, right, here on the predicted age, here in the actual age. Some people look older than they actually are, or they actually look younger. And the question is, for your age, if you now look older than you are, is this a good indicator of risk? Now, they took here kind of the boring example where you can do this for males and females in the association. This is, of course, perhaps not so relevant, but you can also imagine if you would do this for the risk to get diabetes or any other health outcome, particularly of health aging-related outcomes. So this clock concept is very popular, and I just listed a few papers of different omics resources that do that. So also from the organizer and the Rotterdam study, you can do it on transcriptomics. You can also do it on proteomics. And you can do it on metabolomics, a different metabolomics as that we use today. Yeah, so all types of different omics resources can be used to make these clock predictors. So what we have within the consortium in BBMRI, we have a lot of multi-omics data, but particularly we have, together with BIOS, but particularly we have a large resource of NMR metabolomics data, particularly from the Nightingale platform. So a lot of studies now use this. Also, the UK Biobank is now measuring this. Is a relatively small set, omics set. So it contains 226 metabolites, but it's very affordable and it's highly standardized. It's measured in blood, and only a bit of blood is sufficient to measure all these parameters. And what they do at Nightingale is that they, well, try to standardize more and more these measurements so that you also can use some of these measurements as really as for routine diagnostics in the hospital. I'm actually not into this company or also making advertising this. I just find it interesting because for age purposes, you can see that there's a nice age trend in a lot of these markers. So what you see here is from this paper I took here, you see here the different ages on the y-axis, and you see in a heat map depicted for all these different metabolites that there's a strong aging signature. So there's a gradual shift of these metabolite levels with age. And hence you can use this omics resource to train an age predictor. So for this, as previously mentioned, we used the BBMRI data set. So BBMRI consists of a lot of different studies. So each of these studies here is listed, and this is their age distribution. You can see that together, all these different studies have a continuum of the full age of adults. Some studies are small, some are big, some have patients, some are just population representatives. But overall, it's a lot of data which you can use to train such an age predictor. So this is what we did. So we evaluated our predictor in several ways. So for instance, using a 5-fold cross-validation. So basically where you divide the data into 5 equal pieces, you train every time on 80% and you evaluate on 20%. So what you can do is you can train a predictor with a linear regression. So here is the actual age and here's the predicted age. And you can see that we predict this age reasonably well. And also in an independent set, we can now evaluate, and again, it is reasonably well evaluated, predicted. So we used a lot of different predictors to do this, but at the end, we just settled for a very simple model, just a linear regression, because it did a job reasonably well. And in the end, it's also about the difference between the actual age and the predicted age, and I'll come back to that later. So people then also ask me, all the time, like, okay, which metabolites then significantly contribute to the prediction of this outcome of age? Yeah, and just a few warnings here. It is very hard to interpret these contributions. So the previous presenters also mentioned Shep analyses, and I think that would be better to really look at how important a particular feature is. But what you see here is the representation of which particular metabolites contribute what in the prediction of age. So it's here listed by T statistics, and the upper part there are a bit more of these amino acids. Later on, there are also a few lipids. And I think what is important to notice here always is that this listing is very much dependent on which metabolites you actually had in your platform. So if it's very independent and predictive, it ends up here. But if some things are very predictive, but also there are a lot of other correlating features that actually also kind of give the same signal, it might drop in this ranking, but it might still be in itself very predictive. So another way to evaluate whether we do a reasonable job is then to do a leave one biobank cross-validation. So where we each time train on all biobanks except for one, and then project our predictions in the left out biobank. And what you can see from this picture is that, well, to a different extent, but in each of these studies, we do catch an aging signature. So where we do see this correlation between predicted and chronological age. So now, how then do we construct a new predictor or a new outcome variable of interest? So this far, we just tried to predict chronological age, and now we try to construct a biological marker. So a marker for biological age, I should say. So, and how is this defined? As said before, so what you have here is the actual chronological age, and here's the predicted age, and we are actually interested in this discrepancy. So in this case, this person looks actually way younger than his or her actual age. So we think that might be actually protective, or in that sense, an indication that this person is at low disease risk. On the other hand, you might also have people who are looking way older. So in the clock field, this is referred to as delta age, so the difference between predicted and actual age. And typically, since this difference might also very much still relate to chronological age itself, you regress out chronological age to find basically the age-independent part of this discrepancy. So now, how does this look then like? So you want to evaluate, and that's basically every time when you devise kind of a new score, it's only, yeah, well, you need to find out what it actually now indicates, and what, yeah, so basically, does it actually track disease? Different categories. So does it track known risk factors? Does it track current disease status? And we ask ourselves, does it actually track all kinds of future events? Yeah, so that's what we did, and we used several left-out studies to evaluate this. As the first, within BBMRI, we had a lot of different risk factors available. So we used in the whole study the different types of risk factors to see if there's a significant association and consistent over studies for the delta metabolic age, so the predicted age minus the actual age, in all these different risk factors. So you see here a forest plot, you get different studies, the different numbers of people in these studies, and the effects. And what you see, we evaluate this formula. So basically, does delta metabolic age, independent of chronological age, still say something about, for instance, a risk factor as BMI, adjusted for gender? So we evaluated BMI. And what we see is that on average, the heavier you are, the older you look in your blood metabolome. And this is quite consistent. It's a small effect, but it's quite consistent over all these different studies. So we did similar things for BMI, for HSCRP, for CRP, and of course also for the cholesterols, which are also part of the platform. So that's a bit cheating. But also interestingly, we found for antihypertensive medication that also people being on this part of medication look a bit older than they actually are. It is not indicative for the different types of blood pressure, for lipid lowering medication, and gender and smoking. So the next step, we want to evaluate whether it's also indicative of current disease status. So what we used is in the BBMRI consortium, we looked at studies that have at least some cases and also some non-cases to evaluate whether the delta metabolic age associated with this disease state. We did this for two things. We looked at type 2 diabetes and for metabolic syndrome. And in this given example, also here for the diabetes mellitus status, we see a kind of a consistent effect that people who are a case look a bit older in their blood than they actually are. And noteworthy, so here we again associated delta H with the diabetes status outcome while adjusting for chronological age, but also for BMI, because this is a known confounder. If you would leave this out, then the effects would be way bigger. And lastly, we also looked into future outcomes. So on the left, we had a left out study, the longevity study, where particularly in the oldest old, so the 90+ cohort of that study, we looked into survival into the oldest ages. And there we saw that the people with the lowest quintile had the poorest survival, whereas people who actually looked younger than they actually are had the longest survival. So, and here also we evaluate the follow-up time with the Cox proportional hazard model, where we adjust also for chronological age. And to take it a step further, also within PROSPER, which is a study on cardiometabolic outcomes, particularly on the treatment with statins in patients with cardiovascular events, we looked for associations of delta metabolic age with future events like coronary events or cardiovascular events, vascular mortality, all-cause mortality, but also heart failure of hospitalization. And you can see that basically, again, given these hazard ratios, that if you look older in your blood than you actually are, you have a higher risk of getting one of these events. Okay, so to wrap up this part of the presentation, so we are reasonably able to predict age from the blood metabolomics panel. And in that sense, metabolomics clocks do way better in terms of the prediction of age. Transcriptomics do, I think, a bit more comparable. But in the end, it's about this difference between the predicted age and the actual age, and whether this difference is actually informative for disease risk. And there we see that actually it predicts kind of equally well. So, right, so the epigenetic clocks do predict age much better, but then the question is the remainder, the residual between actual age and the predicted age, is this actually informative? And I think these residuals, whether you have a lot left over unexplained or a little bit, is in that sense equally predictive. Now we think that metabolic age might show promise as a biomarker for biological age because it associates significantly with clinical risk factors, with current clinical endpoints, and with future clinical endpoints. And what is a nice aspect is that this thing is trained on highly standardized and affordable data. Now there is of course a lot of interest in different outcome predictors. So as I already said, this work is heavily inspired. A lot of questions. Has heavily inspired on Steve Horvath's work. So he built a methylation clock. Now we are also looking into different cohorts in which we can actually evaluate the methylation clock. So where you predict age using methylation data, and where you predict age using metabolomics data. And what we see is that there's a small correlation. So previously I mentioned here there was an insignificant and non-significant correlation. Well, it depends on the sample size. If you look in large studies, it's also somehow significant, but in the end, it is a kind of a small correlation. And this is the same for a different predictor on the same type of data, which is done by my colleague Joyce Deelen. So he used this data to predict mortality directly. So he trained a predictor for mortality. And basically the discrepancy in age and the mortality predictor also show a small correlation. And this suggests that basically these different types of predictors, either clocks or mortality predictors, are complementary in the information. But then the question is, right, if they show low correlation, now what outcomes do each of these clocks predict? And then also, do they predict these outcomes independently? And this is one of the topics that is studied in this Voila consortium, also with the BBMRI partners. Okay. Another thing for which we wanted to evaluate is what we have within the consortium is a intervention study where we try to intervene on someone's health using a healthy lifestyle intervention. So in short, the numbers of this study, the go-to study, is that 164 people went a 13-week lifestyle intervention where they had less caloric intake and more exercise. From these people, several biopsies were taken from muscle, blood, and adipose tissue. And what we can ask in blood, using, again, metabolomics, whether now if people start living healthier, whether they also improve their the metabolic predictors. And that's actually not the case. So I should say that. But what is interesting is that you can very well train predictors to express who of these people did an intervention and actually uphold this. So you can score the intensity of the effect of these interventions using metabolomics data. So there are different types of readouts, and we think that they might not be informative on each other because this is a relatively short period, so a 13-week change, whereas these mortality and biological age predictors are cross-sectionally trained and on a very long time span. Okay, well, one of the PhD students in our group actually takes now this discrepancy in age to a next step. So what you can also do is look at different types of risk factors that you can predict. So again, using blood metabolomics data in BBMRI, you can evaluate different types of predictors. For instance, for obesity or diabetes status or statin use, and you can use this to, one, impute missing phenotypic data. So showing here for diabetes predictions, we do reasonably well, actually very good. But also you might wonder, so if we again look at this discrepancy or this discrepancy in either diabetes status or obesity, what this actually would mean. So suppose you would be very lean, but in your blood, you would like a very obese person. So I guess this also associates with cardiometabolic outcomes. And this is one of the things we're now currently also investigating. One of the other related activities is together with a student, Kathleen Kuit, and supervisor Dennis Moak. So we looked into different types of platforms, all measuring metabolomics type of signals. So within the Qatar study, there is a lot of different platforms assayed. And basically, we asked the question, suppose you would try to make these clocks or predictors for other covariates, which platform is actually best? And please keep in mind that these platforms are wildly different in the number of measurements that they do, also the costs. But apparently what you can see is that for your particular outcomes of interest, different platforms are more suitable. And what I found quite strikingly is, for instance, that we were not able to predict, for instance, with the lipidizer platform, the BMI status very well. Whereas, yeah, it is on lipids, it is, we think, related to BMI, and it's a lot of measurements. So apparently it captures something else. Yeah, so by comparing these different platforms, we also, yeah, there are different aspects that play a role, like I said, the costs, but also the number of external data sets that you can access. So yeah, these are all driving your choices for the different type of platform that you might choose. So lastly, what we're now working on, so previously a student implemented a web tool where you could download metabol age computations for your own data set. So you could upload your metabolomics data and get metabol age scores back. Now, in the meantime, the paper is published. We're also working on new scores, and Dani is now basically making an R-Shiny application, which you can download, that not only computes metabol age, but also all the other metabolite-based scores, so that you could use this for your own downstream analysis. Okay, this concludes the presentation. Thank you for your attention. These are the people that I would like to acknowledge, and also these are all the funders and funding agencies that make this research possible. Thank you for your attention.
Thank you very much, Erik. Beautiful talk, nice overview. There are quite some questions already, so maybe we can start with the first one from Peter Bram, who would like, oh, now I'm sort of, can you, oh, yeah. So it's
yes,
about linearity, the question. I lost my chat, but maybe you can read it. Yeah.
Okay, Peter Bram, nice talk. Aging is often regarded as a non-linear process. Did you try non-linear approaches? Yes, a lot. And the funny thing is, you can actually do, with more advanced machine learning methods, you can do the age prediction a bit better. But then the residual between actual age and the predicted age is not necessarily more interpretable in terms of disease outcomes. So it's a bit of a, I mean, it's a weird statistical thing where we look at this residual, right? The thing you cannot explain in age and predicted age. But yeah, like I said, these linear models produce highly interpretable scores which do associate with disease. Now, what Dani is also doing is to capture this a bit better, is then also make predictors for different types of age tranches. So middle age, high age, or young age. And indeed, there we see that different metabolites contribute to these different break-off points.
So because I had a very similar question, Erik, maybe there could be like a cutoff of this kind of metabol age or whatever, and then beyond that cutoff, there is a sort of a dysregulation of your metabolic state, and that is then associated with disease. So could you model that kind of concept?
Yeah, so what we're trying to do is now, of course, also, I think like also the previous presenter alluded to, is we try to combine these different scores and then get to new disease states, right? So all these different risk factors are perhaps indicative of particular states. We try to also type people having multiple scores and combine these different predictors into disease predictors.
Okay. So next question. Yeah.
Just because Erik mentioned me, so I just wanted to say that. So we use the age score for actually brain age prediction based on the MRI to show that again this gap is like associated with dementia. But what we tried also, maybe that's for non-linear method, just coming back to this question, we try to use again, like with images, it's only deep learning, but then instead of predicting age, we use that model which I showed for survival analysis. So basically we're trying to directly model the time to the outcome. Maybe that can be interesting to try. Then this you skip kind of step with residuals, and you directly say that, okay, that's associated with risk of certain diseases. So just
agreed. So we have a collaboration also with Newcastle where they try to predict this directly from face images, right? And there, more machine learning is then also naturally involved when you deal with images.
So
just really interesting analysis.
I see a collaboration already entering. Okay, so next question from Alain. What other data would further improve your prediction model? Other omic layers, maybe patient information?
Yeah, so I should then also acknowledge, for instance, other BBMRI partners. So Rick Jansen just recently published a paper where they tried to combine different clocks. So you train on different omics resources, you train and then make a meta predictor on the combined clocks. And yeah, that is one way. I think that's a more natural way. So yeah, of course, you could also concatenate data or try to, think of better ways of making a predictor using multi-omics, sure, yeah.
Yeah, I think the underlying question might be that do the other omic layers capture different aspects of the aging process? Maybe.
Yes, I believe so. And that's also what now these papers show. This is then done in one study. So, I mean, this study has a kind of selection of people, so that you should keep that in mind. But I think more and more when people start evaluating this, that these different age predictors capture different disease aspects. Yeah. Even if they are made on the same data. Yeah.
Okay, so next question from Janine. Have you evaluated the metabol age score in children or adolescents as well? From which age onwards could we use this?
Good question. I have not looked into children. We'll be happy to to look into this. So feel free to contact me about that. No. What I can say, and I think that's also very important, what we now do is we try to use the differences with aging to score people on an aging axis. And this score itself is not necessarily actual age, it's just a relative measure of how old you look. So if you now have, for instance, this children cohort, I can still imagine that some kids are ranked older than they actually are. So it might work, but I'm not sure what a disease for instance, in children really coincides with age-related processes, right? So that's a bit, yeah.
Should be validated then also in that age range, right? Yeah. Okay.
Yeah, or no, I think the best validation is then, of course, go back to your standards in that field of what you think this is disease, this is not disease, this is disease progression, and try to associate these and find out.
Yeah, clear. Let me see. Alida, do you think the intervention could not be predicted well enough as the intervention was not long enough?
Yeah, I think so. I think that is exactly the case. So in 13 weeks, a bit of exercising, a bit of more healthy eating, I think we're not into a new steady state. These people are still in transition, reorganizing their metabolome. And I think what we typically train these predictors on are on cross-sectional data. So where people have been kind of in the same state for ages, and this associates with future endpoints. So if we have a predictor for a poor cardiometabolic state, this relates basically also to their lifestyle and what they do. Yeah, so I think this dynamic process is not well captured by the things we train on cross-sectional data.
No, okay.
But we can actually train these predictors very well on this dynamic data, but it's just a different type of predictor.
Okay. So I actually began with the wrong question. Alessio was the first actually to have a question. It is a long one. It's many of the metabolites showed are predictors of many common diseases, for example, glucose, diabetes, HDL. Is the chronological age a sum of the predictors for the most common diseases? Are the metabolites, lipids associated with overall or chronological health? How do you discriminate between chronological age predictor and specific disease predictors?
Yeah, so what are we capturing? That's basically the question, right? And I think by analyzing the difference between age and predicted age, you tend to pick up age-associated processes, disease processes. But then the thing, your question is, so it's associated with a lot of different health outcomes, this platform, right? The metabolites on this. So do you know, and what do you expect? Well, I think that is a bit of the downside of making these scores. It's the proof of the pudding, right? You need to really show in good longitudinal studies, for instance, that is predictive for future endpoints. So there is really a follow-up step on that. Once you have constructed such a predictor, that you need to evaluate this. And yes, we now see, for now, that it's particularly in the cardiometabolic corner, as you also say with these metabolites.
Yeah. Okay, so there's quite a few messages still. So it's about residual delta H. This reflects actually the variance in H not explained by metabolomics, right? And it is minimized in the least squares to derive the best linear fit. So how does this influence the interpretation of the delta H as being younger or older than chronological age? Do you?
So perhaps you are referring that the residual is still age-related?
Tessel, maybe you can ask the question directly.
Yes, thank you. Yeah, so I was thinking about this delta H and what you exactly capture with it, but you already have been talking about it in relation to the other questions. But I was then thinking, does this not also depend really on the population that you use for your prediction and whether this delta H is indeed positive or negative? So indeed, if the person is younger or older than the biological predicted age.
Yeah, and then you mean in which population you apply it or in which population you train it?
You train it.
Ah, yeah, I guess so. I guess it depends a lot on what type of the population you train it on. In that sense, we also were worried that, I mean, I can understand the thought. We were worried that basically now in this consortium, we have really disease cohorts and we have also population averages. Now, suppose we would leave out all case control settings, or at least all cases, would not now make a very much different prediction. And the funny thing, no, because I think sadly, there's also a lot of, well, type 2 diabetes going on, or cardiovascular problems going on in people who have not been diagnosed yet, right? So yeah, I am sure it will influence a bit of the predictor, but overall predictions were kind of equally well.
Okay. And just out of curiosity, do you have an explained variance? How much do the metabolomics explain of the age?
Yeah, so R squared, you mean it?
Mm-hmm.
0.45, I think the R is 0.65, yeah.
Okay.
It's not that high compared to other omics. Some omics are really good in doing that.
Methylation is really good in doing
that.
Yeah.
Okay.
But like I said, it's about the interpretation of this discrepancy. Is that informative on disease?
Yeah, we don't want to look at calendar age. We want to look at biological age, right? So,
yeah.
So I think last question from Joanna. Not sure whether you want to ask it yourself, Joanna.
Yeah, sure. Hi, thanks for the presentation. I was wondering, so people have asked about the different age groups maybe having a different mechanism here. I was thinking about sex differences, and I noticed that you use gender, well, sex as a covariate multiple times. Do you think it's likely that there's actually a sex difference? So maybe different mechanism or different metabolites being important? And the reason I was thinking about this is mainly because I know that reproductive age, at least in women, also has to do with biological age. So can you say anything about that?
Yeah, so again, we also looked into this when making this score. I'm not sure whether we evaluated on the correct way, but what we did is we trained sex-specific age predictors, right, for males and females separately. We also cross-evaluated them, like, do they reasonably well predict for the others?
Cool.
Yeah, they did. Which is slightly confusing, especially when you say, well, menopause, something really female, so, right, perhaps this is represented data. So what we haven't done, and if I remember correctly, I can imagine if you would really try to predict at this age strata only, from 60 years onwards, that there is perhaps a difference in the metabolites that really contribute to the age prediction for males and females. Right?
Because it's after menopause, yeah.
Yeah, so we just took a very, very broad range from 18 to 100 something, and then looked at what was predictive. Yeah,
interesting.
Good question.
Thanks.
Okay, well, I think we have come to the end of all the questions in the chat. And I think also to the end of this session, I would like to thank all of the presenters for very nice talks, and also the audience for a very nice discussion. Hopefully we see all of you again in 2 weeks in the last of the series.